Hello, everyone. Welcome to the new series for Anatomy and Physiology 2. So, in this series, we're going to be starting with the central nervous system and working through a general overview of Anatomy and Physiology 2, going episode by episode through it. My goal is to make each video, around 15 minutes long. So, there could be a lot of videos by the end of this because there's a lot of systems in Anatomy and Physiology 2. The first section here is on the nervous system. So today we're going to be talking about the different cells in the nervous system, so the different neuroglia, and then we're going to talk about the general structure of a neuron, which is what sends the signals. And after that, we'll go into the action potential in the next video, and then synapses in the video after that. So that's a little summary here for the next couple videos and the one for today. All right, let's hop right into it now. So we're here. There are some basic cell types found in the nervous system. These cell types are referred to as neuroglia. So right here, neuroglia. This does not include neurons. So neuroglia are separated and have different functions based on the central nervous system, or CNS, versus the peripheral nervous system, or PNS. So central nervous system is brain and spinal cord. Peripheral nervous system is any spinal nerve that comes out of the spinal cord. So going to our arms, our legs, our hips, our shoulders, everywhere that's not the spinal cord is the peripheral nervous system. Now there are 4 cell types or neuroglia in the central nervous system and only 2 in the peripheral nervous system. So we're going to talk about the astrocytes, the oligodendrocytes, the microglia, and the ependymal cells, and then we're going to talk about the satellite cells and Schwann cells in the peripheral nervous system. Now, there is some significance in the way I made this table. So astrocytes are similar to the satellite cells, and oligodendrocytes are similar to the Schwann cells. So by the end of this video, we should be able to understand the main differences and similarities between those two. So in the peripheral nervous system, we don't find microglia or ependymal cells. There are cells there that do similar functions, they just don't have that name. So first, let's talk about astroglia and satellite cells. Remember, astroglia are CNS and satellite cells are PNS. So now, what do these do? So these provide installation. They hold things in place. They actually have a lot of very important functions. So they kind of surround the neurons. So we'll show a better image of this later in the summary, but here the astrocyte provides neural cell with physical and nutritional support by cleansing the brain of debris, transporting nutrients, and digestion of dead neurons. So astrocytes are very, very important in the central nervous system. So if I were to draw a capillary here, let's switch to red to show that capillary, and now let's draw the astrocyte coming around. So these little foot-like appendages here actually reach out and wrap around these capillaries. So this controls what flows. Let's say there's another one wrapping around this side. This controls the exchange of nutrients from the capillaries to the brain. And because we don't want everything in the blood getting into the brain. So this is called the blood-brain barrier, or BBB. So the blood-brain barrier is essential in only letting select things in and out. Astrocytes play the key role in that blood-brain barrier by forming that protective layer and controlling what comes in and what goes out. They also form many other important functions. Some of them even make neurotransmitters, which we'll talk about later for some specific neurotransmitters. All right, so those are the astroglia or astrocytes. You'll see them written similar ways and back and forth. Next ones are the satellite cells. Remember, satellite cells are found in the peripheral nervous system. So if we're looking at a cell body, we're going to talk about the different neurons and that we can find in a later chapter. But here, this is a cell body and surrounded by these individual satellite cells that wrap around it. And again, they serve this protective feature. They're insulating, they're directing it where to go. They provide some essential nutrients and so forth. So pretty much holding it in place, and so it can work and can function. So remember, peripheral nervous system, satellite cells, central nervous system, we have astrocytes. And then we're going to have the Schwann cell, which we come up next here. So now we talk about oligodendrocytes. Oligodendrocytes are central nervous system. Schwann cells are peripheral nervous system. Both of these produce a lipid protein structure called myelin, forming something called myelin sheath. Now, each of these do it slightly differently. So an oligodendrocyte provides neural cells with this myelin in the central nervous system and provides insulation in a way. So it wraps these layers, let's switch colors here, wraps these layers around the axons. So this is an oligodendrocyte, and one unique thing about oligodendrocytes is that they can wrap multiple nodes. So individual ones here, these are called nodes of Ranvier, and these are these little gaps. And this myelin is really important to do something called saltatory conduction, which we'll talk about later, where the signal can jump from node to node to node and travel very quickly. So how fast an impulse travels down an axon is determined by both the extent of myelination from in the central nervous system, specifically from oligodendrocytes, and then also the axon diameter plays a role in how fast the signal travels. So smaller the axon, the slower the signal, the more myelination axon has, and the wider diameter, the faster the signal, like the nerves running up our sciatic nerve. So those are oligodendrocytes, and this is a nice image here showing the wrapping of that myelin. So let's switch to black here. So this myelination here, out here, it doesn't show the cell body too well, but so the oligodendrocyte reeks out, and this is just the membrane of the oligodendrocyte comes out and just wraps. It continues pushing around and around, circling, circling, circling. As it pushes the cytoplasm, or the fluid inside the cell, outwards. So the cytoplasm then is out in here. And there is very little cytoplasm in there, forming this dense layer of lipids and proteins. So it's like the rubber on an electrical cable, insulation, and provides protection for those axons. And so now, and faster conduction. So oligodendrocytes are found in the central nervous system. Now, a Schwann cell is slightly different. Schwann cells can only wrap one individual section. So this would be one Schwann cell. Out here, it's not drawn, would be the cell body, also known as the neurolemma. And here, this releases the compounds, and they continue the wrapping function. So every Schwann cell has its own cell body and neurolemma. So one big difference is Schwann cells can only wrap one section. It's still myelin, but it only wraps one section compared to oligodendrocytes, which wrap a bunch of sections, like in the central nervous system here. So there's one oligodendrocyte wraps 1, 2, 3, 4, 5, all these different sections here. Whereas a Schwann cell, it's 1-to-1, so wrapping one node at a time. And that's really important in the peripheral nervous system, and actually allows some regenerative capabilities in the peripheral nervous system. So this neurolemma can interact with that one and kind of repair this region in between if there's damage. And also, same thing, we have the nodes of Ranvier here, and you can jump from node to node during saltatory conduction, which is really important. So Schwann cells, peripheral nervous system, oops, I'm still in black, peripheral nervous system, oligodendrocytes are central nervous system. But both of those provide insulation and increase the speed of signals in each system. Next cell type here are the microglia. These are only found in the central nervous system, and they serve an immune function. So they're very similar to macrophages. So macrophages are these cells that go out and they engulf foreign particles. They bring them in and then destroy them. So in our brain, we have cell debris from leftover neurons. These microglia go out and digest those dead neurons. And also, if there are foreign particles that enter the brain, which should be prevented by that blood-brain barrier, so these microglia are the immune system of our brain and spinal cord that help protect it and break down cellular debris so they can be reused by the cell. But these are similar to macrophages found throughout the rest of the body. So now, the last cell type here found in the, again, central nervous system are the ependymal cells. So they are found right here in this layer on the outside. And on these layer of cells, we see this layer of cilia. So these help move what's called cerebral spinal fluid, or CSF. So out here is the fluid-like substances is moving around these cilia beat, and it helps move it through the brain. So these ependymal cells are only found in the central nervous system. Now we do have ciliated cells and other mucous membranes in the body, which we'll come back to in other chapters, but this is the important ciliary cells found in the brain. More of these are found in a region known as the choroid plexus as well. So moving through here, we now get finally to the neuron. So this is a general structure of the neuron. This is called a multipolar neuron. There are two other types of neurons. One is called a unipolar, and then we have a bipolar neuron. So a unipolar neuron kind of has a cell body here that pokes off the middle, like that one we saw earlier with the satellite cell, so it looks more like a T. So down here then we have the axon terminals, and over here then we have the dendrites. These are found in the dorsal root ganglion. So here, let's draw it a little more like that. So the DRG, so our sensory neurons have these. And also the effectors also have a ganglion as well. So we see these types of neurons. Again, these are called unipolar neurons. This neuron in this image here is called a multipolar neuron. And then the other one is called a bipolar neuron. So a bipolar neuron has dendrites here, has a axon, has a cell body in the middle, and then the axon here, and then synapses down there. So these ones are found in vision and smell. So we'll come back to those when talking about the special senses for vision and smell, and we'll see these bipolar neurons come back. And here they have that cell body in the middle. So this is axon, whereas this unipolar here, this is axon, cell body comes off of that like a T shape. So this is more like a T in a way. So again, these are found in the sensory neurons mostly, and also any ganglion that we mentioned in the brain, they don't have to be unipolar, it's just a collection of cell bodies. So when we look at now, our focus here is the multipolar neuron. So here, multipolar neuron, this is called the axon. It's the long segment that runs down through here. That's surrounded by the nodes of Ranvier right here, surrounded by the myelin sheath. And here you can see how every individual neurolemma here is represented. So remember, Schwann cells. Each one is a separate cell. Now over here we have the nucleus of the cell body. Cell bodies, when we have a cluster of cell bodies, is known as a ganglion. Then from that cell body, we have what are called dendrites. These are all these appendages sticking off that are receiving signals. So we might have signals coming in from neurons here, neurons here, neurons here. So there are going to be thousands upon thousands of dendrites that are synapsing. Well, thousands synapses that are synapsing on this cell body, sending the signal. Or depending on the receptor. We'll talk about the different kinds of receptors coming up later then, but this is where the signal is received most of the time. Then the signal travels down through the axon, again, jumping from node to node to node. We're going to talk about how that conduction works in the upcoming videos, and we get to the end, which is known as the axon terminal. The axon terminal is where then we form synapses, and we send that message to where it needs to go. So we have to be able to direct that message from one cell to the next and keep it intact. And the next cell then summates that information, determines whether or not another, what's called action potential, is going to be sent. This is known as the axon terminal. And we'll talk about that a lot more in the next video or two for how the signals are sent. But you can have tens of thousands of axon terminals. That means one signal coming through this neuron could then go tens of thousands of different ways, or it could go one way, or it could come backwards. There are a lot of different things that could happen, a lot of different circuits that form in the brain and in the nervous system, depending on it. So this is just a general representation of a multipolar neuron. It's the most common neuron found in the body, especially in the central nervous system, and very, very important. So that was just a quick summary of the different glia, neuroglia, and a quick summary of the different kinds of neurons that are found. So we talked about the microglia, which are the immune cells, the astrocytes, which hold neurons in place and also direct the transmission between the capillaries and control what comes in and out of the brain. So we have our astrocytes there. The ependymal cells have the ciliated structure here, moving the cerebral spinal fluid around, and then the oligodendrocytes are forming these myelination around individual axons. Remember, one oligodendrocyte can form multiple, whereas one Schwann cell can only form one around the peripheral nervous system. So this is a summary of the central nervous system here. Remember, peripheral nervous system astrocytes are replaced with the satellite cells, and oligodendrocytes are replaced with the Schwann cells in the peripheral nervous system. So I hope this was a quick, brief summary of all the information, and I hope you got a great take-home message, and I hope you all have a great day. Thank you. Hello again. Thank you for watching my video. I'm glad you could come by. If you enjoyed this content, feel free to do that thing where you like and subscribe. Also, if you're interested, I prepare and record all of these videos live on Twitch. After the recordings, we do a post-recording question and answer section, and then sometimes I play games where any question is fair game as I game. Also, on Twitch, I'm a co-founder of a wonderful community called the Knowledge Fellowship, or TKF. Our goal is to seek and share knowledge with others while live streaming. We feature streamers who cover biology, law, physics, chemistry, geology, photography, health and fitness, psychology and mental health, rocket science and aeronautics, programming, and tons of different practical EDU streams. Feel free to check out TKF at the link in the description and join the very active Discord community. Hopefully, find something that interests you. With that, I'll see you all next time and have a wonderful day. Remember to seek knowledge and challenge everything.